My name is Hannah Lather, and this summer I did my internship at Dr. Tamer Salam's lab at UCLA, and my mentor was Elena Sanchez, and my project would be on the characterization of DDX17 in macrophages. So before I begin, I'll be going over the clinical relevance and why DDX17 was our focus. So first of all, atherosclerosis is a very prevalent condition that can lead to cardiovascular disease. It is caused when cholesterol plaques build up in the artery wall, causing an inflammatory state. Examining these inflammatory pathways can be meaningful to see new therapies for atherosclerosis. DDX17 is an RNA helicase that is involved in multiple RNA processing pathways, and recent research has shown that DDX17 can act as a sensor detecting certain RNA and activate inflammasome complexes. Inflammasome-driven inflammation is a very major contributor to atherosclerosis. Therefore, studying DDX17's role in the immune system, particularly in macrophages, can be important to see if it has a role in the inflammation in macrophages. So our hypothesis was macrophage DDX17 plays an important role in atherosclerosis. And our objective was to create a DDX17 knockout in macrophages and examine their response to inflammatory stimuli. I'll be moving on to the methods. So first, how do we achieve the DDX17 knockout? First, with the crate mice, we insert a lyse M crate, which is a macrophage-specific crate. And for the LOXP mice, we tag the target gene, which would be the DDX17 gene, with LOXP insertions. And in this gel electrophoresis of the DNA, you can see that in the wild type, in the lower bands, these will be the DNA without the LOXP insertions. In the heterozygous, they'll be the bands with LOXP insertions. And as you can see in these sequences, these will be the LOXP insertions basically. And when we cross the cray and LOXP mice, we get the mice with the conditional DDX17. So this will be a little techniques overview. I start with the cray positive and cray negative mice. And to clarify, cray positive would be the cells without DDX17, and cray negative would be the cells with DDX17. And I would incubate them in different inflammatory triggers. And then we analyze the DNA with qPCR and the proteins through Western blot and ELISA. I'll be moving on to the results. So these are the different pathways that my inflammatory triggers will have. First, one of the incubating triggers would be poly-IC, which is a double-stranded RNA that mimics a viral infection. Binds to the TLR3, does a phosphorylation cascade, and will release pro-inflammatory cytokines such as IL-6. And the LPS is mimicking a bacterial infection. It binds to the TLR4, does a phosphorylation cascade, and will release pro-inflammatory cytokines such as TNF-alpha and IL-1 beta. So these are the qPCR results for the poly-IC incubation. The red is representing cray positive and the blue is representing cray negative. And first, for every experiment, we have to make sure that we have a good DDX17 knockout. So if you look at the bar graph on the left, you'll see that the cray positive has a significant less expression of DDX17 compared to the cray negative. And here we can see that there's a trend in which the cray positive has more of these inflammatory markers, inflammatory RNA markers of inflammation. Therefore, maybe cray positive is experiencing more inflammation than cray negative. And this is a Western blot analysis of the proteins, comparing cray negative and cray positive after the LPS incubations. And as you can see, these are proteins involved in the inflammatory pathway, PP65, PERK, PJNK. You can see that the bands are darker for the cray positive side compared to the cray negative side, which means that the cray positive has more of these inflammatory proteins, therefore is experiencing more inflammation. And then we have another Western blot with different proteins. This time in the top we have the protein caspase 1. And as you can see, pre-negative and pre-positive have similar concentrations, but only in the pre-positive cleaved caspase 1 cleaved, we can see that there's more of these proteins cleaved caspase 1, cleaved gastrin D, pro-iolate beta, in the incubations for LPS and ATP. And it's showing that it's following this pathway more than it is than for the pre-negative. And the ATP incubation was needed to activate the inflammasome. And this is an ELISA analyzing the IL-1 beta, which is the pro-inflammatory cytokine. And this side is the standard curve. And these top 4 were the pre-negative, and these bottom 4 were the pre-positive. And as you can see, there's some saturation here, but it wasn't the result we expected to see. And like this was ideal, but the technical replicates didn't follow the same result. Therefore, we were thinking that maybe there was a lack in ATP incubation, and we'll try to repeat the experiment this time with a longer ATP incubation time. But still, we see a similar trend in which the Cre-positive is experiencing more pro-inflammatory cytokines compared to the Cre-negative. So for future steps, we can have an RNA-seq analysis. We can repeat the study with anti-inflammatory stimuli. We can do a cholesterol overload incubation, or we can do an in vivo test, which is like a real-life mice test, and afterpharmacy study. And I would just like to thank everyone in the Salam Lab. They were so friendly. They were always saying like good morning, good afternoon, things like that. And my mentor, Elena Rodriguez Sanchez, and of course the CIRC program who gave me this wonderful opportunity. Thank you so much. Any questions?
Yeah, that was great. So I guess just a question. It looked like you were measuring cytokines at the 1 hour and the 4 hour
Yes.
time point, and there was certainly a lot more present at the 1 hour time point. But why did you have those 2 time points, and was that what you expected, that there was more at the 1 hour than at the 4 hour?
So we actually expected the cytokines to be more released after 4 hours, because a longer incubation time, we're thinking there's going to be more an inflammatory response. But in the ELISA, it wasn't very ideal, and we're thinking it's because of a lack of ATP incubation, because it was like 15 minutes, and it should have been much longer, so it can actually follow through the inflammasome activation.
So they're going to do that, just like you said, in the future with longer ATP incubation and redo
Yes.
this. So,
Yes.
So for the Cre-positive and the ELISA, were you expecting more yellow like coloration in the plate?
Exactly. So the yellow is indicating that there's the cytokine that we want, and if it had more of the IL-1 beta, it would have had a much higher concentration of the yellow color, and it would have been ideally this color. You can see it's much lighter. Therefore, we're thinking it's the lack of ATP incubation that causes it.
But was it intended to be like in all 4 of the wells, or just those on?
That's a good question. So we expected it to have more for the Cre-positive, but of course for the Cre-negative as well, because it should experience some sort of inflammation. You can kind of see there's a lighter yellow in these, but not as much as we expected.
And then was there a difference between like the 3rd well and the 4th well, or were they the exact same thing?
Yeah, so in the 3rd column and the 4th column, there will be technical replicates, and then by row, it's different timings and incubations. The 4th row being the LPS ATP with